Vincent Hilser. I'm the chair of the biology department, and I'm a professor in the department as well. Biology as a discipline is very broad, and I deal with the molecular mechanisms of how protein molecules work. So proteins are obviously one of the sort of major molecules in cells, and I study sort of how they work. And in particular, I study how information, so we know that proteins are not just structures, static structures, they sort of undergo conformational changes as part of their function. In fact, those changes are the basis for all function. And how they go about these movements and the implications of how binding drugs or binding the receptors affects other parts of the molecule, which allows them to interact with other things. That's what we try and study, because it's at the heart of really all biology. We use methods of calorimetry, which is a small set of things that allow us to measure thermodynamic properties. And you know, at the heart of all biology, of all physical processes, is thermodynamics, but that's a very small set of methods. Mostly what we use is spectroscopic methods, and they can be NMR spectroscopy or you know, different forms of light spectroscopy. Things like circular dichroism and fluorescence and ultraviolet and visible spectroscopy. So those are the sort of at the core of the science that we do. I think you have to sort of get an idea of what the sort of challenges are in biological systems in all sort of science. So the physical sciences are really about observing phenomena. So you have to be able to see it. You have to be able to, if something happens, you need to be able to see it. Unfortunately, physical science is predicated on the behavior of molecules, and those aren't too small to be seen. So we have to take advantage of the properties of these molecules. And so molecules are made up of protons and neutrons and electrons, and that allows us then to perturb them with energy. And the energy that we perturb them with is light, so of different wavelengths. So in order to see them and see certain aspects and properties of them, we use spectroscopy. So depending on what we want to see, we employ that type of spectroscopy. So it's really, you know, spectroscopy is the key to us being able to observe them. And you know, you could sort of almost boil science down into studying developing sort of ideas about how things work and trying to find ways to actually see it. And spectroscopy is really the venue, the avenue that folks, that allows us to see things. Because we're interested in looking at a range of processes and phenomena and properties of proteins, we use a range of spectroscopies, and we use them in a couple different ways. So one particular spectroscopy that we use is circular dichroism spectroscopy. And what this is you just take advantage of different absorbance properties of proteins. So proteins, as we know, are made up of chiral molecules. And chiral just meaning that they have a handedness to them. They have mirror image properties, as we learn in our general chemistry. If that's the case, then that means if you have circularly polarized or different types of polarized light, the different handedness of the molecule will absorb different ways. So dextro or level rotary, if you absorb differently, then you can get certain properties of proteins, structural properties of proteins, by looking at the CD. So one of the things that we know that when protein molecules fold up into their conformations, they will absorb or have a certain signature if they're exposed to this sort of polarized light that is sensitive to this. And so, for instance, alpha helices, a structure that's that's common in proteins, has a certain characteristic absorption property with polarized light. So we can look to see how structured molecules are under certain conditions. So it's just a general technique that allows us to understand whether it's structured, and then if you look at that property as a function of adding another molecule that it binds, or destabilizing it with temperature, or seeing what effect lowering the pH has on it, you can see that change in that property with those conditions. And you know, over the years, the field has learned how to analyze that information and, you know, determine a lot about it. You know, how stable is it? Does a mutation to this as a result of a disease, does that destabilize? What's the origin of the disease? We get at the molecular basis of the disease, and that molecular basis is usually when something happens, you know, there's a difference, a genetic mutation, that will affect a physical property of the protein, either its stability or its ability to interact with something that's critical to function. And that's what we try and do. We try and first identify what the physical basis of that is, and we can use spectroscopic techniques to do it. And then, you know, if we're lucky, identify ways that we can remediate that, either to design another drug to, you know, stabilize it, or, you know, genetic therapies, all sorts of things that are, you know, much more complicated and much more in-depth. One of the things that we sort of apply all of the techniques that we're looking at, you know, calorimetric techniques and spectroscopic techniques of different kinds, is we're interested in a class of molecules called transcription factors, and these are things that turn genes on and off. And the class of transcription factors we're looking at are the class called steroid hormone receptors. And they're important because in that family of proteins is estrogen receptor, and glucocorticoid receptor, and progesterone receptor, and androgen receptor. These are all, you know, if they don't ring a bell, they should ring a bell, and that what they are involved in is significant amounts of cancers. Androgen receptor is prostate cancer, and estrogen receptor is breast cancer. And misregulation or dysregulation of these molecules is associated with these types of cancers. So we have to know how these things work, and, you know, how the mutations or you know, how whatever is going wrong with these is related to cancer. So this is what we're studying right now, and it turns out there's a whole, you know, class of protein behavior that has been discovered in the last 20 years, and that's these sort of sequences that are what are called intrinsically disordered. That is, the fluctuations that I study, that we sort of, you know, allude to in biology, where you have structured molecules that sort of move around a little bit around an average structure. That's important. But these kind of molecules, they have no unique conformation. They're basically flopping all over the place. So they're really difficult to study. But, you know, to understand why people get cancer, we have to understand that process. So we're working on two particular versions, androgen receptor and glucocorticoid receptor. We're trying to figure out the relationship. What about these molecules and the differences between these molecules in different individuals allows some people to develop cancers and other people to not. So, you know, if we know about this and we know why, then can we develop intervention strategies? So that's, you know, so we've, you know, taken all of these techniques, spectroscopy being, you know, forefront among them, to, you know, go in there and interrogate these systems and figure out how we're going to, you know, make a difference at a you know, medically relevant level.